A very warm greetings to all the viewers. In the previous lecture, we discussed about characteristics of immune response and branches of immune response, which are humoral mediated immune response and cell mediated immune response, where we were talking about antigen presenting cells. We will talk about one of them today, and that is micro mantra for the day, dendritic cells. Hello and welcome. I am Micro Wizard, Dr. Dhruvil Brahmbhatt, working as an assistant professor in Department of Microbiology in LG Institute of Applied Sciences situated in Ahmedabad. Dendritic cells were first discovered by Rolf Steinman and Zhenwil Kohn in mid-70s when they were studying spleen cells to understand the induction of immune response in major lymphoid organs of the mouse. They were awarded with Nobel Prize in 2011 for their work. Dendritic cells constitute approximately 0.2% of WBCs in the blood but play a very important role in non-specific resistance. They are present in skin and mucous membranes of the nose, lungs, and intestines, where they readily contact invading pathogens, phagocytose, and process antigens and display foreign antigens on their surface. This process is known as antigen presentation, or also known as professional antigen presentation. The cells which perform this function are known as antigen presenting cells. These dendritic cells are critical for initiation of immune response. So they are both a part of innate immunity and also work as a bridge between innate and acquired immunity. They are named so because of the long branches which are similar to dendrites of neuron, which extend and retract dynamically, and hence they are named as dendritic cells. The question is, where do they originate from? The dendritic cells is developed from either lymphoidal progenitor or myeloid progenitor in the bone marrow. Now let's try to understand the role of dendritic cell. Several barriers of our body, let's say skin. Just beneath the layer of the skin, there are dendritic cells, macrophages, which lay underneath the skin and wait for pathogens. Now let's assume that there is an invasion of foreign particle or antigens or pathogens. These invaders are immediately recognized, identified, and an attempt to eliminate them by cells such as dendritic cells is carried out. These dendritic cells recognize specific pathogen associated with molecular patterns, also known as PAMPs, that is pathogen associated molecular patterns on microorganisms. These molecular patterns enable dendritic cells to distinguish between self and foreign antigens. Once the pathogen is recognized, the dendritic cells pattern receptors, that is PR, the dendritic cells pattern receptors bind the pathogen and phagocytose it. How do they do this? Well, the receptors send signals into nucleus which leads to cytokine formation. The cytokines are released at the site of an invasion, which helps to combat pathogen efficiently. Dendritic cells, which are phagocytosing cells, digest the cellular proteins and present on the surface of the dendritic cells, which migrate to lymphoid tissues, where as activated cells, they present antigens to T cells with the help of MHC2 complex. Antigen presentation triggers activation of T cells, which are crucial for initiation and regulation of effective specific immune response. Thus, not only do dendritic cells destroy invading pathogens as a part of innate response, but they also act as a bridge and present the antigens to specific cells for effective immune response. I hope this video was helpful in understanding the role of dendritic cells as antigen presenting cell in our body. Do hit the comment section below for doubts or suggestions. Take care, keep reading. Thank you.